want to use some type of conjugate because he originally knew that he was going to do the
like some
antibody.
I have no idea what using experimental therapy. When is the 70 week form? Oh, 20. I can actually submit it now already. Can you
Finish it?
finish it on my second day?
Oh, you will?
I want to get all the assignments out of it. By this I tried. Today is on acetylcholine or no. That means I didn't download class.
Okay, so we're going to start. So the online student, you can hear me. Yes, okay, good. Good, thank you. So last week, we stopped at the kinase linked receptors. So these are the group of receptors for different hormones, growth factors, and they actually have a tyrosine kinase domain at the intracellular side. Okay? Some of those hormones, for example, cytokine released by the immune system. That actually can have also activate the cytosolic kinase in the kinase linked receptor. Basically, this group of receptors, they have a extracellular domain. They recognize the hormone growth factors, and then they have the intracellular enzyme links to it, the tyrosine kinase. Now, the receptors actually they share a common architecture with extracellular, as I mentioned, the ligand binding domain, and then there's a single helix go through the membrane, and then have the intracellular domain. Okay. The intracellular domain have a characteristic tyrosine kinase over there. So this group of receptors actually are very important in the neurons and also in different cell types as well. But this one is actually pretty important. Particularly, support the neuronal survival. Okay, particularly nerve growth factors that we will have some examples in the end. Okay, the signal transduction is binding of the ligand and then cause the structural changes, and usually will involve the dimerizations of the receptor. So receptor binds to the ligand, change the structure a little bit, and then they form a dimer. Once that they dimerize that, and then that will cause the autophosphorylations by the enzyme, the tyrosine kinase, at the tyrosine residues. Okay. Now after it's phosphorylated, then that will cause the change in the structure, and then there will be a protein with SH2 domains binds to the receptors. And then once it binds to the receptors, then it will trigger the secondary messengers. And then that actually will control the cell growth or differentiations. Okay, in the neurons, particularly, there will be support for the survival, and also in differentiation, for example, the neural outgrowth, or help to form, additional synapses and so on. So here's one of the example growth factors. So there will be recognition site, the transmembrane helix, and then the tyrosine kinase residue domain. And there are some tyrosine over there. Okay. So once that it binds to the ligand, the conformational change will cause induced by the ligands. Then that will form the dimers. Okay. Once they form the dimers, then the enzyme domain will phosphorylate the tyrosine residue. So it's now phosphorylated, and then the SH2 domain protein will recruited to the many background noise and echo. Let me change the mic a little bit. Is it better? Is it okay now? Okay, so we'll continue. Maybe I will speak up more because I don't used to have the mask to speak, to teach. I'm getting used to it. It's getting better. Okay. And first day. Okay, so now once the SH2 domain protein binds, and then that will trigger the activations of the secondary pathway, and then that can cause alter the gene transcriptions and then tissue response. So we will talk a little bit more about those like this type of receptors compared to the other one, the anotrophic, metabotrophic, the G-protein coupled receptors, and then this one the kinase linked receptors. Now one idea why the phosphorylation will cause such a difference in the protein for receptors. So usually phosphorylation will occur in serine, threonine, or tyrosine residue. So in this type, this one is tyrosine.
Actually, the question was,
so why phosphorylation will change the characteristic of the protein?
it's a genetic charge of the protein.
Yes,
Yes,
it changed the charge of the protein because the tyrosine is hydrophobic one. If you add a phosphorus group on top of it, then you got a charge, then that will change its binding character. So here is what happens. Okay.
But then why does it require dimerization for phosphorylation to happen? Like why can't it happen independently?
Well, because if they don't come together, they cannot do the autophosphorylation. They each have the the kinase domain.
Oh, okay.
Good. Okay. So some of the example in this case, this one is the growth factor binds to it, and then that recruit the GLB2. This is the SH2 domain protein, and then it binds to it, and then that will phosphorylate the SH2 domain, and then that will recruit activate the RAS, and then the RAS will go on the RAF MEK and MEK kinase, and then that leads to the gene transcription. Now, because of that will support it for the cell divisions or growth or differentiations. Okay. Another very common type is the cytokine in the immune system. So cytokine will bind and then recruit the JAK domain, the phosphorylated, and then after phosphorylations, the SH2 domain protein, for example, in this case, the STAT will recruited and phosphorylated, and then that will cause the dimerizations and then go for the gene transcriptions. Okay. Now that actually will cause a lot of the changes in the inflammatory response and so on. Okay. Now the protein phosphorylation in signaling, we'll talk a little bit about it. Those actually will mediate a lot of different processes. So protein phosphorylations, for example, in other secondary messengers, that will induce protein phosphorylations that change their properties, either sometimes activate the protein sometimes they will control the receptors, sometimes will control the channels, and so on. So that's a very important in the receptor-linked tyrosine kinase case. Now that involves the cyclic nucleotide activated tyrosine kinase and other kinases as well, and involve the kinase cascades. Now those ones that it integrated into secondary messengers, then that you integrate together depend on the combined effects. When we talk about a little bit more, for example, the cell is not just activated by, or neurons, not just activated by one type of receptors. Now, the G protein-coupled receptor, when it is activated, that will also cause the secondary messengers cascade as well as does the receptor-linked tyrosine kinase receptors. Okay, so those needs to integrate together, and then that will have an output. Okay, so there are many kinases, very different substrate. And actually, those have been studied quite well in terms of the different phosphorylations. And in other classes, you may have heard about those as well. Protein phosphorylation in the signal transduction again. Now, that can also actually can desensitize the G protein-coupled receptors, as we will look at it in the receptor regulations. That can cause the internalizations or enhance their function, depends on which type of the modification on the receptor. Now, apart from the kinases, there are also a large family of phosphatases as well. So that actually remove the phosphoric group and then deactivated the protein or reactivated, depends on the process. Now, phosphatases actually has not been studied so well compared to the kinases for some reason, because it's more difficult to study for the removal of the phosphor group. And the reason behind is actually if you activate something and then that activate the kinase and then the kinase will phosphorylate the substrate, that's easier to study. Okay. Now, phosphatase is rather is different, but it's just remove the phosphor group. But how it is being activated and how it's being controlled is less well studied. So if you look at some of the papers, and usually it's more study on the kinases. Okay, for some of it, when the time we talk about the secondary messengers, you seldom see that actually there are phosphatases over there, because some people might think that there are non-specific phosphatases to remove the phosphor group, and that's the reason why that actually not more specific one has been identified or studied. Okay. Okay. So now we are going to the intracellular receptors. So this group includes a lot of steroid hormones, the thyroid hormones, vitamin D or quinonic acid. Those are the ones that actually are hydrophobic. And then they can diffuse across the membrane and then binds to the target, okay, the intracellular receptors. Those receptors are protein, so the ligand must first enter the cell first, okay. Now then you see that actually this class of actually receptors or hormones will have a more global effects. Actually depends on the cell type, which we will talk about in one of the examples that you may have heard about in the treatment of the COVID-19 recently, that you might have heard about it. The receptor consists of a conserved DNA binding domain attached to the ligand, and there's a transcriptional control domains. Because this group of receptor usually will affect the transcription level, and will have a long-term effect on the overall, not just in the nervous system, but it will have a global effects, and that's the reason why it is very powerful hormone that you might have heard about in some of the cases. Now, the DNA binding domain recognized specific base sequence so that it can promote or repress particular genes depending on the sequence and also where is the cell type. Okay. So as I mentioned, the pattern of the gene activation depends on both cell type and nature of the ligand. So that will be highly diverse. So that can actually, for example, alter the protein synthesis, and those can be slow in onset, but have a more long-term consequence. Okay. Some of those, the nuclear receptor is responsible for the increased expression of some of the drug metabolism. So sometimes that actually can induce the changes in the enzyme and the drug metabolism. Now, this is one of the examples. This example is the famous glucocorticoid steroids. Okay, this is a steroid that has been used in the treatment of the both in the SARS and also in the case in the COVID-19 recently. Now, in this case, for example, the glucocorticoid steroids will diffuse into the cell, and then that actually combines to the receptors, and then displace, in this case, the HSP90, and then after that, they can go into the nucleus and cause the effects. Okay. Now, from some of the example of it, that is another example in which that in some cells, the initiation of the synthesis of protein can be initiated by the transcription factors. Okay. However, if there is a, you can see that in this case, if there's a glucocorticoid receptors binds the activated and binds to the transcriptional effectors, and that actually can induce less protein transcriptions. In this case, related into the immune cell. Another example in which that it can bind and the glucocorticoid responsive elements in the DNA, and that can actually initiate the synthesis of the anti-inflammatory protein. Now, in this case, the glucocorticoid in the system actually is immunosuppressive. It is actually also a so-called a stress hormone. When people get stressed, that actually will release this type of hormones. When we talk about depressions, then we'll discuss a little bit more of it. This is a very powerful steroid. So why in the SARS and COVID-19 they will use this kind of immunosuppressive treatment? Now, even the famous US president is taking this class of drug as well. Anyone?
Is it like the COVID-19 is related to the autoimmune response, and having this glucocorticoid steroid kind of reduce the reaction?
Yes. So one of your students, if you cannot hear, actually he said that suppress the autoimmune response. So usually when the immune system find out that there's some new things coming out, then they'll get really stimulated, and then we will release a lot of cytokine and different anti-bacterial or antiviral chemical or molecules. Now, in the SARS case, that is actually very obvious, because the SARS actually involved a much higher immune response. So that's the reason why the SARS is much easier to contain, because most of the people getting it will get fever. So that's the most easy. But you can see that now this COVID-19 actually is more difficult, because it's not everybody could get fever. And then you can carry it around and then you don't even know about it. So that's the problem of it. So sometimes if the virus is too lethal, actually it's more difficult to spread. But if it's not that lethal, actually it's more difficult to contain, just in the case like this. So maybe until that we get the vaccine before we can go back to normal. Otherwise I don't know when we will resume to fully face-to-face. Okay, now this is for the immune system, but actually steroids, even glucocorticoids actually can affect the nervous system. Okay, not just in the immune system, but also the nervous system as well. The effect is less well known, but we know that actually it can suppress some of the dendrite development. Now, other parts of the steroid, can you think of anyone that actually can have a very obvious effect on the body? That you have it, I have it. Some people purposely taking those. Nothing of any online student. Yes, good. Testosterone. That's the male hormone, and also the female hormone as well. Now you see that those actually can change your actually in some cases change the even for the thinking. Now there was a study on those stock brokers in Wall Street or stock exchange. Now, usually they find those traders actually have higher testosterone. Very interesting. You know why?
They're highly stressed all the time.
Well, they should have stress hormone. Too much money. Not necessary. So some people think that actually because they are more aggressive actually. Now when you do trading, you have to be aggressive before you make money. Some people are saying that. So that's the reason why that if you have watched the movie, what's it called?
Wolf of Wall Street.
Wolf.
Wolf of Wall Street.
Yeah. You see that those people are taking a lot of cocaine.
Oh, yeah.
So you know they are rich as well as those make them make some risky move actually. For those people that I hope that you are not taking any of those or trying any of those. Those study has shown that those people taking those actually they make some more risky decisions. Okay. So those are the steroids. Now you see that actually this is true. That the steroids actually have profound effects on your nervous system and make you change your thinking sometimes. So those are studied or previous history show that if you abuse testosterone, particularly in the athletes, there will be a long-term effect on your thinking and your body as well. And at the same time, you can see that testosterone not just change your thinking, but also change a whole category of different effects. For example, make you build stronger muscle, make you have enhanced endurance. That's the reason why those athletes are banned from taking testosterone as a supplement. Okay, so it's banned in, for example, Olympics or swimming because that the hormone will have a very wide effects. And you will see that actually the hormone, the steroid, will have different effect on different cells. Okay, depending on the type of cell. So time scale of different receptors. Now there are some times the receptor needs to quickly respond to the environment. Sometimes it needs a longer response because you need to change the cell characteristic of the cell in the extent to change the long-term effect. Okay. So the time elapsed from the binding of the agonist to the ligand-gated channels will be very fast. Okay, that will be milliseconds. It's very crucial because that, from moment to moment to transfer the information across synapses. For example, the movement, how fast you move determine what you can do. Okay. For example, in the case that the animals needs to escape from the predator, then you need to move very fast. You see, so that's the reason why that actually those needs to be very fast. Now, however, in other mechanisms, for example, that may require minutes or even hours, for the case that in the gene active hormone, for example, in the corticoids, the effects can persist for hours or even days, depending on what drugs. Now, this has the implications on the use of drugs for the treatment of different diseases. For example, in the treatment of the acute bronchial asthma. Now, asthma is actually a very serious condition. People actually can get killed by it. Okay, in a very serious condition, particularly in other cases, asthma or allergy can also can also result in a serious effect. It's not very hot. It's not very. The asthma is actually common in Hong Kong. Actually, I hope that none of you are being affected by it. If you unfortunately suffer from it, just be careful. Actually, I know of classmate in high school. Actually, he was in his 20s and he died because of asthma. Just suddenly one night that is cold and then that relapse of the asthma. Now, in the treatment of the asthma, maybe you have seen it before. It's like a aspirate thing that you suck up into the trachea. Now, actually, those contain drugs that for both two major ones. One is the adrenergic receptor agonist. So that actually release the symptoms at once because that adrenergic receptor agonist is acting like the adrenaline. So when adrenaline is released, your bronchial muscle will dilate it, so that will help to release it. Okay. Now, when we talk about the noradrenaline, then you'll see that this is actually a localized effect because if this is a more general effects, then they will have a different outcome. Okay. Now the other one is glucocorticoids. Okay, that will have a long lasting effect, more than 30 minutes now or days, because actually that will help to suppress the immune response in the bronchioli. Okay. Now actually the allergy actually is something that also affecting a lot of people. For example, the skin and so on. For some of the really serious allergy, for example, in US in particular, some kids actually they are allergic to the nuts or peanuts. Okay, those actually are they need to be very careful. Even a small amount of those peanuts or nuts will cause a acute immune response that can kill them actually. That's the reason why if you look at some of the nuts that you can buy in the supermarket, actually they will claim that they cannot guarantee that is not contaminated with nuts or peanuts. Okay. So that's actually a very dangerous condition, even though it's not very common in Hong Kong. Very strange. It's kind of have cases in the US or Western countries. So here is the integrations of the molecular and cellular mechanisms. So here is the anotrophic receptors. So that will cause the hyperpolarizations or depolarizations, and that will last for milliseconds. So really quick response. The other one, the G protein coupled receptors, that can also link to the iron as well, but that will be much slower because that involves the activation of the secondary messengers. Okay. For the other one, involving calcium ions, Well, I think that is, I think the effects actually happen when some Congress did. So why is it happening now, but not in every widespread cases? So it's a step that actually those children must have exposed to some other things that trigger that actual allergy to happens in those cases. Maybe living style, because I have heard that some kids that living in the Western countries, and then when they come back to Hong Kong, they get to develop it. But in Hong Kong is much less common. So it must be the lifestyle, or maybe, yeah, you're right, maybe it's genetic. It's not necessary. The world want them to go away. It's not necessary by that, okay? So the other thing, protein phosphorylations, and then T cell response. For the hormone, for example, the steroid hormones, that will take hours or even days or weeks before that the outcome will be obvious. So that's the reason why that actually the athletes, they have to take the steroids if they want to cheat. They have to take it for a long time, and that's also the reason why that actually the Olympic staff will check on those famous athletes from time to time to see whether they are taking the drugs. Okay. Okay. So receptor regulations. Now, when we talk more and more, you will see that actually the receptor and the nervous system in particular, or actually all the cellular system, is not unchangeable. It's always changing. Particularly important is in the nervous system actually, and that allows the system to respond to changes in the environment. Okay. So that's the reason why the receptor, for example, the amount is not constant. It can go up, it can go down, so that the system can fine tune to respond the best to the environment. Okay. So how is it being regulated? This is one of the examples. This is the beta adrenergic receptors. So we can see that there's a G protein associated with it. And then if the agonist binds and then activated, we can see that the receptor is responsive. Okay. Now, however, if it's always being activated, then the system says that maybe we need to down-regulate the response. So there will be the receptor will be phosphorylated, and then another protein called beta-arrestin will bind to the phospho group in the receptors. Then the receptor, then even the agonist can bind, and it will not be responsive. Okay. So until some time phosphatase kick in, the phosphorylated group, and then the G protein can active that can interact with the receptor again. Then the receptor will be responsive again. Is it okay? So just want to remind some of you again that I haven't received all the group formations. I have received 6 groups so far, and if you haven't formed your groups, please do so as soon as possible. Otherwise, I will need to form it for you. Okay. So just to remind you that if you haven't sent me the group formation, please do so. Okay. So receptor regulation again. The receptor needs to be at the cell surface before it can function, particularly the anotrophic, metabotrophic, and enzyme-linked receptors. So actually, the receptor at the surface is a dynamic process. Now it can be endocytosis and then destroyed, degraded. Okay. Now they can be internalized by endocytosis, and then stored at the vesicle level. And then when it is needs to go to the surface, then that will go to the surface again. Now they can remain on the surface, but change so that they cannot bind naked. Okay, some of those. And or they can bind naked, but will not form the receptor ligand complex that does not induce a normal hormone responses like the previous one, that phosphorylations. So all have that can upregulated or downregulated, depend on the stimulations or activation of the pathways. So sometimes the receptor signal transduction can be enhanced, and sometimes that can be downregulated. Okay. Now that actually by the end of this sections or the receptor sections, then there will be some examples that you can see, and in the further other classes in this course, we will have a more examples to show that how actually that will be regulated. Okay. So we'll talk some about the secondary messengers. The activated receptor will induce a response in the target cell, either directly or indirectly by the production of the secondary signal, called the secondary messenger or second messenger. Now that target different response or also different cellular mechanism, metabolism, or even in the gene transcriptions. Some of the examples that you might have learned in other classes, cyclic AMP, GAG, or P, triphosphate, triphosphate. Now, the hormone binding to a G-protein couple receptors can activate the phospholipid C, phosphatidylate, inositol 4,5 diphosphate, PIP2. Okay, so that's how they found the IP3 and DAG. So one of the important aspects, actually, of the secondary messengers can magnify the signal transductions. You can see that the integrations of different molecules, for example, from the receptors to the G protein, to the cyclic AMP and protein kinase, all the way down, and then that can amplify the signal. Okay, so that's the reason why that the secondary messenger is more powerful. That's also, you can see that the chemical synapse is much more powerful than the electrical synapse, because that the integration of different secondary messengers that can enhance or cancel each other. Okay, for example, the GI and GS and GI G-protein coupled receptors, they can cancel each other because one increase the cyclic AMP, one actually decrease the cyclic AMP. Now, this is another example of how that they can integrate together. The receptors have to activate the G protein, then they can activate the adenyl cyclates or phospholipid C, and all those integrated together means the effectors can be enzyme transport proteins, contractile proteins, okay, or ion channels. When the ion channel has changed, then the cell can be more sensitive or less sensitive to the stimuli. For example, if the baseline of the cell is lower, then a smaller activation can trigger the cell to have a strong signal. On the other hand, if the channels actually decrease hyperpolarized the membrane even further, then actually the cell will be less difficult to activate it. Okay. Then actually, have been shown that in particular in the synapse, some synapses are more responsive, some are less responsive, because that in the case, some of the secondary messengers can change the overall baseline of the neurons, and then they can be more responsive or less responsive. Okay, that's actually based on the memory formations in a sense. Okay, for example, one of the ways to change it, for example, if you are focused, now, just for the case, we'll talk about it in the memory formation as well a little bit. If your attention is stronger, your ability to remember will be much better. So that's been why if the class can attract your attention, and you think about it in more, you can remember it better. You know. Now that's the same as your daily exposure as well. You know, like if you have, well, if you have a boyfriend or girlfriend, those things happen between you and her, or you and him, will stay in your memory much longer. Well, not good things, bad things will stay stronger as well. So if I ask you what is your favorite restaurant, you remember. If I ask you what is the worst restaurant, you also remember because you have that experience in that restaurant. Okay. Same thing, like just like on campus, maybe you miss the restaurant or you don't miss the restaurant here. But some of those are really bad, and then you have bad experience for that. You remember, and you will try to avoid that. This is actually some of the survival technique. Okay. So that's the mechanism behind that. Actually, the mechanism, yes, it's like fear, disgust, or anger. Memory linger longer. So that actually is a protective mechanism for you. Okay. When you enjoy more, you want more. If you something make you scared or fear, then you also will remember. Okay. Like if you hurt yourself by somebody, so you remember that you will avoid it. So there's a saying, fool me once, shame on you, fool me twice, shame on me. Because if you have been fooled by twice, if you still don't remember, that's your problem. Okay, so that's the indication. But now this is some example. But when we talk about it, then there's some mechanism actually enhance the formation of those memories. That's the reason why when we talk about the different nuclei, I mentioned that the amygdala control the fear, but it also control the formation of the memory as well. Okay. So calcium regulation. Now, calcium regulation is very important in the nervous system. Well, however, it is also important in different cells as well. Well, it is particularly important in the nervous system because that actually can control the survival of the neurons, actually. So in many aspects, the calcium actually is bind to the calmodulin before the calmodulin will bind with some other component and activate it. Okay, for example, in the nitric oxide, cases generation in the neurons that involve the calmodulin. When we talk about nitric oxide, then you will see actually. The intracellular calcium is determined by calcium entry, calcium exclusion, and exchange in the cytosol and the pathway reticulum. Now, actually, in the cell, in the neurons, the calcium level is kept actually in a low amount. So one of the calcium importance that you have learned about it is in the synapse. Still remember, the reduction of the calcium actually will cause the release of the neurotransmitters. So you can think about if a lot of calcium is going into the cell will release a lot of neurotransmitters. So either it will be good or it will not be that good. And when we talk about the neurotoxicity, actually you see that how calcium can cause such bad idea. And one of the cases is in the stroke. In the stroke, a lot of cases actually involve calcium toxicity or excitotoxicity. Okay. Now here is the diagram that you can see how different calcium levels being controlled. So the calcium can enter by 4 different routes. Okay, so one is the voltage dependent calcium channels. So that is in the synapse in which the actin potential arrives. Change in the membrane potential can activate the voltage gated calcium channels and will result in the open. Okay. Now there are also some of the ligand gated calcium channels as well. Okay, you'll see that there's some acetylcholine, for example, glutamate or ATP. Acetylcholine that will be related to the, for example, muscle contractions or other consequence of the activation of the neurons. Okay. So there are store operated calcium channel as well. You can see that over here. Now, how the calcium is being excluded? So we can by the pump to exclude the calcium either to the cytosol or to the endoplasmic reticulum. Now, endoplasmic reticulum actually is very important in the storage of the calcium. Okay. There are channels of myelin receptors, for example, allow the calcium to come out. Now, particularly in the muscle contractions, you might study in the physiology. During muscle contraction, the acetylcholine will go to the neuromuscular end plate, and then once it's accepted, the nicotinic receptors, and then allow the calcium to rush in. At the same time, to activate the endoplasmic reticulum to release the calcium, so that to enhance the muscle contractions. Okay. So that's how that actually, when there's intracellular signaling, for example, in the case of IPV receptors, that can enhance the calcium to release out and increase the outside calcium that can activate different pathways as well. So here's some of the example of the calcium secondary messengers. There's calcium, the sources, for example, endoplasmic reticulum, and also in the voltage gated calcium channels. Okay, there are targets. You can see some of those targets over there. Cyclic GMP, cyclic AMP and cyclic GMP. You can see that the sources and then the targets. So those are actually protein kinase A, protein kinase G. Then that can actually get different kind of gauge channels changes. There will be also the activators. You can see that phosphodiesterase that can remove the cyclic AMP, similar in the cyclic GMP phosphodiesterase. The IP3, there will be phosphatase, and then different enzymes. So here is an example of diagram that shows similar to the previous one, how the calcium is being controlled. Okay, from channels to the endoplasmic reticulum, and one of the things for the calcium is actually the mitochondria as well. Okay, which is also very important during the case of the stroke actually. Now, just give you some a bit of idea. In the stroke actually is during stroke there will be not enough energy because of the blockage of the blood vessel in the nervous system or central nervous system. Now, without the energy, the membrane potential will reduce it, and then that will trigger the opening of those a lot of channels. And then when the calcium is too high, then that will be have a bad consequence for the neurons. Okay. Here is the example of this cyclic AMP signaling. This one is the phosphatidyl inositol biphosphate pathway. You generate the dioxide glycerol and also the IPV in the signaling. And then that is actually another one is the nitric oxide. Nitric oxide is actually important in the neuronal signaling as well. It's being synthesized actually by different type of the nitric oxide synthase. If you have to learn it, there actually in human there are 3 types of nitric oxide synthase. When we talk about nitric oxide signaling, then we'll talk about them. Actually, it's very important in the neurons and also in the circulations and so on. Now, this is one of the examples of the nerve growth factor that has been studied quite importantly. This is NGF, so the nerve growth factors. And then when it binds to the TRAP-A receptors, which is the tyrosine kinase receptors, they will trigger it to form the dimer, okay, because of phosphorylations. And then they will activate different pathway, PIP kinase pathway, WAS pathway, and PLC pathway. Now that involve the cell survival and also neuroid and neuronal differentiations. Okay. Now this is actually very important, particularly during your development. Now, when you are still young, or even nowadays you are still not fixed yet. What happens? The neurons actually going out and then make connections. Okay. Now the connection will form only if they support each other. Okay. So here is how they encourage, for example, the release of the growth factors, so they support the neurons. And then once they get support over there, then the connection will become stronger and become more stable. Okay, and that's how they survive. So once that you get to more mature, your network kind of more likely form that cannot regroup or be modified. Okay, so that's the reason why that if you have maintain a good development or learn something well when you're young, once the network is formed, you remember it even when you are getting older. Okay. So one of the examples is that actually, for example, I always, well, when we talk about memory, then we will talk more, but one of the examples is like learning to ride a bicycle. So once that you learn it, then you cannot forget it, actually. You may not be very good at it, but once that you try again, and you get it back. Because that actually get into your solid memory. So, for example, if you know how to ride a bicycle, you know that you don't need to look at the wheel. You can just balance it automatically. I still remember the time that I learned how to ride a bicycle. You always have to look down to the wheel. Same for you if you have a driver license. Anyone that have a driver license? Not yet, huh? Yes? Not yet. So if you have the experience in learning how to drive, so you see that actually you have to always, particularly if you are learning how to drive the manual stick, you always have to look at the stick and then try to see where it is, what gear actually it is. But if you are driving automatic, that will be easier. But in the future, actually, maybe you don't need to do that anymore because more modern cars actually is not very, particularly for example, electric car actually the gear is actually not that important anymore. Okay. But that's the thing that when you learn, you have to look at. That's the reason why when it is forming those networks. So that depends on the growth of the synapse and development of the synapse, or connections, how strong it is. Okay. So here is another example of how that the secondary messengers can affect the outcome. Now, this is one of the examples called the long-term depressions, in which we will talk about it in more detail in the memory. Now, when the neurotransmitter release that binds to the metabolic glutamate receptors, this is the case when it is not strongly excited. Okay, that actually binds to both the ionotrophic, in this case, the AMPA receptors and the metabotrophic receptors. Now, the signal will come in, but if it is activated not strong enough, the metabotrophic receptors will have the secondary messengers, and then actually we will affect the AMPA transmissions, and then that will let it less easy to activate the postsynaptic neuron. So that will have the so-called long-term depressions. This is an example that if you have a repeated weak excitation, then you actually depress it. I always give an example in which that your mother, your parents, like saying very gentle to you all the time, ask you to come back to home for dinner more often, then you will ignore it. But if your mother sounding really loud and then say, if you're not coming back for dinner, I will not cook anymore, then you will come back to have dinner once or twice. And after a while, and then you might forget again. So when we talk about the memory, the long-term potentiations and long-term depression, we'll give an introduction on how those mechanisms are involved, in which actually involve the secondary messengers and also protein synthesis. Okay. So this is also another example of what happens a secondary messenger related to it. Now, if this is in this case, this is a catecholamine terminal. Now, when the action potential arrived, calcium rushing, and then that will trigger the release of the neurotransmitters. Okay. However, at the same time, it will also have a secondary effect as well. So the calcium will also activate it, because calcium is also a secondary messenger. That can activate the protein kinases, and then protein kinase can phosphorylate, for example, in this case, tyrosine hydroxylase. After phosphorylations, the tyrosine hydroxylase will activate it, and then will produce more of the catecholamine. Okay. So the reason behind is actually it can have a feedback so that the terminal knows that it is active. So it needs to produce more neurotransmitters to enhance the subsequent activations or release of neurotransmitters. Okay. So this is a fast way to compensate if the synapse is activated. Now, but if this is continuous being activated, then the cell might produce more protein, so that to compensate as well. Okay. Now there will be other ways to do it as well. If this is too active, there might be auto system, the secondary messengers, and then to modify the sensitivity of the terminal, so that if you need much stronger stimulations before the cell will release the neurotransmitter. So you can see that actually both ways. So that's this some of the mechanism of the habituations or stimulation or sensitizations actually. Okay. So any questions regarding the receptors? So if not, then maybe we will stop here a little early, because I don't want to start the coding sections today, because that might be a little bit too short if we end at in 5 or 10 minutes in time. Any questions? So just to remind that, 19th of October, we will have the midterm. I'm still making the exam questions, and I'll give you more informations later. Now the midterm will cover up to today's lectures. Receptor. Okay? Is it okay? Why you are so happy?
There's less stuff to remember.
Oh,
I still need it for final exam.
Yeah, so you will find it. Anyways.
yes. So up to today's lecture, I know it's not cumulative. If I want to do cumulative, you will not happy. During regular class time, and this is conduct online, because we don't want to have too many students together. Yes. Regular class time. Is it okay? Oh, okay, wow. Open book, I haven't decided yet, but you want open book? Open book will be more difficult. Huh? I will finalize it when I finalize the questions. So one of your students asked about the metabotrophic receptors are being blocked for certain reasons, and that will be long-term potentiation. Well, actually, this one is here is a little bit simplified because we in the diagram that didn't include the another receptor, the AMD receptors. Yes, but the metabotrophic receptor certainly will contribute to the development of long-term or long-term potentiations or long-term depressions. Okay. Okay. If no more questions, any more questions? If no, then we'll end here. Okay. 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 Goodbye. See you on Wednesday. Okay. Bye bye.
Oh.